That feeling when you finally scratch an itch. Pure relief. Deeply satisfying. Almost addictive. We've all felt it. But what happens in our brain at that moment? Neuroscientists in Belgium found something fascinating. They identified a stop scratching signal in our nervous system. When this signal malfunctions, consequences can be severe. Itch is a complex sensation. Once seen as a minor annoyance, it's now recognized as unique. It has its dedicated neurons, pathways and circuits separate from pain. When an irritant hits your skin, like a bug bite or allergen, immune cells unleash chemicals. These chemicals activate itch-sensing neurons, called prurireceptors. They send a signal to your brain, telling it, itch. Instinctively, the brain responds and commands, scratch. But what happens at the molecular level? Researchers at the University of Louvain investigated a protein called TRPV4. This protein is an ion channel found in sensory neurons. Ion channels act like gates that open to stimuli, like pressure or temperature. TRPV4 helps detect pressure on the skin. Researchers initially expected TRPV4 to help with pain, but they found something unexpected. It played a crucial role in regulating scratching behavior. They engineered a genetic mouse model to study TRPV4's specific role. They deleted TRPV4 only in sensory neurons. Then they induced chronic itch in these mice. Surprisingly, without TRPV4, mice scratched less often. But when they did scratch, each episode lasted much longer. Mice with intact TRPV4 scratched more frequently but shorter durations. They found relief quickly and moved on. It might seem paradoxical, but it reveals critical insight. TRPV4 serves dual purposes depending on its location in the body. In skin cells, it triggers the itch response. In sensory neurons, it detects pressure from scratching. This feedback sends a signal to stop scratching. When TRPV4 functions properly, we scratch and then stop. Without it, like in those mice, scratching becomes compulsive. Understanding this mechanism explains why scratching feels satisfying. It's a biological signal indicating enough pressure has been applied. That signal leads to relief, creating a feedback loop. Itch, scratch, pressure detected, relief signal, stop scratching. When this loop functions correctly, scratching resolves efficiently. When it fails, it can lead to chronic itch conditions. Chronic pruritus affects millions worldwide, causing unrelenting distress. It often arises from conditions like eczema, psoriasis, or nerve damage. Traditional treatments focus on triggering the itch. But this new research raises another question. What goes wrong in the mechanism that should stop it? Without the TRPV4 stop signal, relief is elusive. Scratching continues, leading to skin damage and inflammation. For drug developers, this discovery is a game changer. Current treatments often try to block TRPV4 completely. While that reduces itch, it complicates stopping scratching. A completely blocked TRPV4 means fewer itch instances, but harder relief. Future therapies should target TRPV4 based on its location. Blocking it in skin cells while preserving its function in sensory neurons could be key. That way you can reduce itch without losing the ability to stop. TRPV4 is part of a broader family of TRP channels. Each responds to different stimuli, heat, cold, or pressure. Interestingly, TRPV1 enhances TRPV4's activity during certain itch responses. This interaction adds complexity to our understanding of itch signaling. Research is uncovering how these channels talk to each other. Another factor in itch is a protein called ATF4. ATF4 manages itch sensitivity and interacts with TRPV4 directly. Mice lacking ATF4 itch more, while those with more experience less itch. This insight offers another avenue for therapy. It can help fine-tune the body's natural systems instead of overriding them. For millions suffering from chronic itch, this research is vital. Current medications like corticosteroids don't help everyone. New biologics have emerged, targeting specific itch signals, yet none tackle the stop scratching part of the equation. The next generation of therapies should balance these functions. Understanding how we scratch and stop teaches us more about our bodies. The interactions happening at a cellular level are wonders to explore. Detecting when to stop scratching is a critical function. For many, relief is on the horizon thanks to this research. If this newfound understanding intrigued you, share the video and follow our channel for more fascinating insights into everyday science.